Yes, thank you for the introduction. Good morning, everybody. So we'll switch to antibody discovery space. So maybe first one introduction about MImAbs. So MImAbs is a small company in Marseille, dealing with early stage discovery of monoclonal antibodies. We are now part of a larger group, Janvier Biosciences Group, together with other companies in the service space, Edelris with small molecules with the same positioning compared to MImAbs. And then a series of companies with animal models, in fact, PDX, genetically engineered mice, and also different models of mice, trying to better understand the pharmacology of our products. And now there are big synergies between all these companies to really have a better characterization of the antibodies that we produce. Coming back to MImAbs only, so we are positioned in the antibody space, early stage discovery of those antibodies, and pharmacological profiling of those antibodies so that they are compatible with the future pharmaceutical developments. So for a given target, we begin by generating a collection of monoclonals, and we characterize them both biochemically so that they are compatible with future development, humanize them if needed, and make some pre-CMC work to better characterize their biochemical behavior. And in parallel of that, we test those antibodies in vitro for cytotoxicity, for example, or blocking capacity, and in vivo in mice models. So at the end of our work at MImAbs, we deliver a candidate that is really compatible with pharmaceutical development and a package of pharmacology data that ensures that there is a therapeutic window for the combination of the antibody and the target. And then this is taken back by our clients, biotech and pharmaceutical companies that make the GMP development and finally go to the clinics. So the quality of our work, in fact, is attempted by the capacity of those clients to go to the clinics, and we have the chance that several of our clients, on the basis of the work we have done on several targets, have gone to the clinics. The last one that we generated together with Emergence Therapeutics, for example, was on Nectin-4, so that's disclosed antigen that we generated monoclonals and makes antibody drug conjugates and all the pharmacology. It was taken back by Emergence to GMP development, and it was after that taken up by Eli Lilly, attempting that what we have done at the early stage preclinical level is compatible with even the requirements of big pharma companies. So what we do to do that, so the first step to begin is really to have a good collection of monoclonals, because of course you have to screen that for all these characteristics, both biochemical and also pharmacology. So you really have to start with a good collection of antibodies to be able to select a good candidate for future development. So the way we did that in the past was to rely on mice immunization, because usually we have a reasonable collection of monoclonals of good affinities using that procedures. And we use the hybridoma technology, so that's a very well-known technology for antibody, fusing the myeloma cells with myeloma, and then screen for recognition of antigen. So I must say that it's a robust technique, well proven, and we have had several successes with this technology. But it has some advantages that I mentioned, but also some drawbacks. First, in terms of speed, because you have to wait for the hybridoma to grow and then sequence them and then produce them recombinantly and then work on them after that for immunization and so on. So it's quite a long process. And also it works well with, let's say, good antigens, but for difficult antigens such as GPCR or ion channel, for example, the depth of screening using this technology, we can screen about 1,000 hybridoma at one time. So for those difficult antigen, we had several failure to generate good antibodies for those particular antigens. And we wanted to have a technique that could be more rapid, and also to allow more in-depth screening, interrogation of the antibody repertoire of the immunized mice. So we switched to the Beacon technology and the antibody workflow on this machine. So it has been presented already several times during the previous sessions, so I can go relatively quickly on this. So you recognize the machine with 4 nests, and you can put some chips on those nests, and each chip has what we use is 11K nanowells. So you can, for each chip, interrogate 10,000 plasmocytes in our case, and as there are 4 chips, that's also 40,000 possibilities of screening in that. So it's really a machine that allows to grow the cells and to maintain them in culture, but also to position them with a light beam. And you've seen that already. So you can square the individual cells and push them in the nanowells so that you can interrogate them after that with several assays. So in our case, we load plasma cells, so from immunized mice. So we isolate plasmocytes using kits based on CD138 expression. And we load that on the upper channel of the chip. And you see on the right that you can square the different plasmocytes above the wells and push them individually in the wells. So that's a very rapid procedure, about 30 seconds. And you have more than 90% monoclonality of your plasmocytes in each well, again, with the possibility to interrogate about 40,000 plasmocytes for a single campaign. So after doing that, you can load on the upper channel assay, different types of assay. So you can load beads, for example, coated with antigens, or transfected cells expressing your antigen of interest. and you can put that with a fluorescent secondary antibody that will signal the positive plasmocytes within, let's say, assays between 30 and 40 minutes usually for a single assay. So this is a fluorescent signal there that you see here. So what happens is that the plasmocytes secretes the antibody diffuses, and then hits the assay beads or transfected cells, and the secondary antibody focus on those beads or transfected cells and signal the presence of a specific antibody for this particular antigen. What is interesting also is that you can do serial testing, so with either other beads or other transfected cells, particularly if you want a cross-reactive antibody on different species, for example, you can load transfected cells with other species. So you can interrogate first in 40 minutes the recognition of the antigen, the human antigen, and then check for cross-reactivity or other functional assay on the same well, so that at the end you can recover a multi-parametric characterization of the antibody secreted by a single well. So after that, you spot the interesting monoclonal antibodies. So it's a procedure that is within hours instead of course, the long growing of hybridomas. And you can upload the interesting plasmocytes on the channel, and then you wash the channel and you direct your plasmocyte in 96 wells, for example, individually, and then you can recover by single cell PCR the VH and the VL of each plasmocytes. So there are kits to do that, to first sequence VH and VL before having the chemokine, and so we could spot different epitopes after immunization with the peptides on the chemokines, different epitopes after immunization with the peptides on the chemokines, intracellular peptide domain or extracellular peptide domains, either with immunization with recombinant proteins or mRNA. And in that case, we counter screen on the machine. So we first screen on transfected cells with the antigen, but counter screened with either empty cells or cells that were expressing cynomolgus membrane expression, for example, to test for cross-reactivity. And in all of those cases, we had really very good numbers of sequences recovered from those different campaigns. And what is really interesting here, so that it's much quicker and wider compared to the classical hybridoma technology. And we have the sequences first before going into tiny production experiments, so that we can select on the basis of the sequences of the CDR, for example, the ones that are the most interesting, and discard those who had problematic CDRs, for example, in terms of amino acids that are affecting, for example, the stability of the antibody. But in a way, for those classical antigens, the procedure is quicker and allows this wider screen. But we had also some cases where the Beacon machine allowed to get a good collection of antibodies, and we couldn't have that, any of them, with the classical hybridoma campaigns. So the first one is MHC plus peptide combination. So we immunized with MHC plus peptide with the idea to have a specific antibody against the combination of MHC plus peptide. And we can do that, isolate specific antibodies with hybridoma procedures. But on the machine, we could counter screen on the beads coated with MHC plus peptide and the same MHC with a near-relevant peptide. So over several hundreds of antibodies recognizing the MHC unspecifically compared to the with regard with the peptides, we could counter screen on those who were really specific of the combination of one MHC plus one peptide, and we had 12 antibodies here that were really specific for the MHC plus peptide and did not recognize the same MHC with an irrelevant peptide. The two other ones that I wanted to discuss are two difficult antigen, one GPCR and one ion channel, and this is the next slides of my presentation. So the first one is GPCR. You know that those are difficult antigens, 7 transmembrane with very tiny extracellular domains. We made several hybridoma campaigns with immunization with transfectants and didn't have any good results with those. And so we decided to really compare hybridoma versus the Beacon technology. So that was the first difference. And the also other difference was the way we immunized the animals. We started to immunize with mRNA instead of transfected cells because there was no recombinant antigen available for this particular GPCR. So that's the results of the immunization of the mice with the GPCR, either transfected cells on the left or cells plus mRNA on the right. So you have the different titers obtained from the sera of immunized mice against the transfected cells. So that's done by flow cytometry. And the lower panels show the absence of reactivity of the sera on untransfected cells. So we had good specific titers, and you observe on the right that you have much more good specific titers with the immunization combining cells plus mRNA. And in the next slides, with mRNA alone as immunogen on the left, you do see that immunizing with mRNA alone is sufficient to have very good titers on those difficult antigens. On this difficult antigen, very specific, of course, using this type of immunization with really no background on untransfected cells from the sera. And another advantage of this technology is that we could also immunize with cynomolgus mRNA. So we alternated between immunization with human mRNA and cynomolgus mRNA to have more chances to get cross-reactive antibodies for the two species. And there was no antibodies to our knowledge for this particular target that had this cross-reactivity with cynomolgus. So with that improvement in titers, with those different immunization schedules, we started to compare, in fact, between hybridoma technology and the Beacon technology. So that was the same mice that were used for the two campaigns. So here we did two relatively big campaigns with mice immunized with mRNA or cells plus mRNA. And we screened about 3,000 hybridoma for those two campaigns. And from those two big campaigns, we had only one specific antibody recognizing the specifically the GPCR target, which is better than before, but still a very limited number to engage into a selection of a good candidate. So we took the same mice immunized with the same antigens and decided to compare with the Beacon technology. So we isolated the plasmocytes from the immunized mice, and we made two chips loaded with plasma cells coming from the cells plus mRNA immunization, or two chips, and in parallel, two chips loaded with plasma cells from mRNA only immunization group. And we screen on the Beacon on the transfected cells, either BAF3 or CHO, transfected with GPCR. So here, well, maybe it's not very visible here, but it's an overview of an old chip, 11K chip, and you distinguish already compared to the hybridoma campaign that there are several specific spots on the whole chip, and on the right you see that you had different kind of signals, either quite bright and some were a bit more fainter. But on those different chips, we had between 20 and 30 positive blooms on the 4 chips. So we could start with quite a good collection of plasma cells that we could export and sequence. So in fact, initially we from experiment 1, which was mRNA immunization, experiment 2 with cells plus mRNA immunization, we had 46 blooms and 36 blooms respectively. And at the end, we, of course, with a single cell PCR procedure, we lose some material. But we had 41% recovery of VHDL for experiment 1 and 72% for experiment 2. Usually you get between 60, at least in our hands, between 60 and 80% recovery of VH and VL, depending also on the number of tests you perform on the machine and the lifespan of the plasmocytes. But usually can recover between 60 and 80% of paired VH and VL from the Beacon machine. So we decided to produce that. So there was several antibodies that were produced from both groups. So on the left here you have the EC50 on transfected cells from different antibodies isolated from mRNA immunization plus cell, and on the right mRNA only. So you see that mRNA immunization is sufficient to have a very good collection of antibodies with quite good EC50, and the same for cell plus mRNA. On the lower panel, you see that we have several antibodies that cross-react on the cyno version of the GPCR. So that's also EC50 obtained on the cyno transfectant. And we had, in fact, 3 antibodies that had similar titers compared to similar EC50 between cyno and human in this collection. So instead of one positive candidate here, we had about 40 different antibodies with good or reasonable EC50 for this antigen, and we had 3 cross-reactive antibody against cynomolgus. Also, in terms of functionality, you know that the GPCR epitope is very tiny extracellularly, so usually the antibodies are not blocking, are blocking antibodies for their ligands. So we decided to test that. Also, we had fluorescent ligands for this GPCR, and we made a competition test with this fluorescent ligand on the transfected cells and see if the antibodies were blocking or not. And you see here that we had 2 antibodies that clearly were non-blocking. And again, it's to our knowledge, it's the first time that there are some non-blocking antibodies that were available for this particular target. So again, quite a good collection of antibodies, and also functionally and in terms of cross-reactivity, a greater choice possible and allowed by these new campaigns. And they are pursuing their, we selected some of them, and they are pursuing their development with our clients. The next example, I would go quicker now, is an ion channel. So again, it's a difficult antigen. That's a tetraspan antigen, multimeric, very tiny extracellular loops. And we decided to make a beacon campaign on these. We immunized again with mRNA and cells. The titers were more modest and with more non-specific staining compared to the GPCR I described before, but we had some mice that gave quite significant specific titers on the transfected cells against the non-transfected one, the green one here on the right, for example. And so we decided to make these campaigns. We used 4 chips, also isolate plasmocytes, and made the screen on the transfected cells with this ion channel. And you see here the final results after production of, so we isolated in fact 30 blooms on the machine for this antigen. And we produced all these, we recovered the sequence of those and produced them recombinantly. Of course, we've taken some very faint blooms. So in that case, we didn't have all the antibodies recovered that recognize the target, but we had 12 antibodies that clearly recognize the transfected cells that are on the bottom panel here compared to the non-transfected ones. So we are reasonably sure that the antibodies are specific. I must say that the transfectant that we used, as we had no control antibody here, the antigen was transfected with a tag. So we wanted to see if the antibodies here are really specific of the antigen and not of the tag. So we had some mRNA for this untagged mRNA for this target. And we could check with our recombinant antibodies that after transfection with untagged mRNA, you have recognition of the transfected cells compared to the non-transfected one with this untagged mRNA. So we are really sure that the antibodies are specific of this ion channel. So to our knowledge, it's for this ion channel, it's the first antibody available worldwide. So to finish, and as a conclusion, so the beacon procedure and antibody workflow really increase the depth of screening and diversity on classical and difficult antigen compared to what we can do with hybridoma. You can have a functional screening at the single cell level of recombinant antigen and transfected cells. So sometimes you don't have recombinant antigen. So you have to really rely on transfected cells. And what is very important for even classical antigen is that you have the sequence before going into the production. And I must say that most of the time when you have a clear bloom on the machine, it translates 100% of time in an antibody that do recognize the target. So the only exception was the ion channel, but we really selected very faint blooms in that situation. So you sequence only the pre-screen hits. You can look at the sequences when you have a lot of those sequences to discard those who have problems in terms of CDR. And again, it decreases times from hit identification to recombinant production, about 45 days to make the whole process to obtain purified antibody for those collections. So with that, just want to make some acknowledgements of what we call the beacon team at MImAbs. So we had a dedicated team for working with the machine, and of course all the molecular biology and production was already available at MImAbs to facilitate all the production. And with that, I will finish. I'm happy to take questions.